Howdy, folks. So this is our lecture 15. Seems like I probably won't be on campus today because our family is going to be getting a little bit bigger. But I want to teach you some stuff about DNA, and I think this should work okay. So I've got these cartoons on the screen that are sort of what you might see about DNA if you hear about it on TV or on a TV show or something like that. And this kind of ridiculous picture that somebody might show you if they were talking about GMOs, genetically modified organisms. But this picture is really nothing like what GMOs are really like. So we're going to learn some cool stuff about both of these today. First, DNA. DNA is a polymer, so it's kind of like some of the polymers you've learned about already this quarter. But it has some big differences. One, it has 3 different kinds of monomer. So it's got phosphate pieces, these things they call sugars. Deoxyribose is not quite a carbohydrate, but pretty close. It looks a lot like glucose. And then these bases, they call them, just because they've got nitrogen atoms in them, which are kind of basic. So there's 4 of those that could make up any one chunk of DNA. So all those are sort of the building blocks. Those come together to make a nucleotide. So when we put together phosphate, a sugar, and one of those 4 bases, that makes a nucleotide. And that's pretty much a monomer of DNA. So we've got these nucleotides. And if we put together multiple nucleotides, here we've got 1, 2, 3, 4 nucleotides, then we can start to see a piece of DNA appearing. So we've got the sugar, a base, phosphate. There's sort of a backbone made out of the phosphate and the sugar, and then the bases are dangling off the side of that. Okay, so DNA has a direction, so the 3' end and the 5' end, they call it. That comes from the way chemists have agreed to assign numbers to the carbons in the sugar. So this is the 3 carbon, this is the 5 carbon, this is the 5' end because this phosphate is connected to the 5 carbon, this one's connected to the 3 carbon. When DNA is copied in your body, it's copied from the 3' end to the 5' end. So it has sort of a direction to it, which most polymers we've learned about definitely didn't have. So there's some differences. When we go and look at DNA a little closer, so this bar graph on the left actually comes from someone grinding up samples from humans, fruit flies, corn, and some kind of mold. So they ground up the samples, they used some kind of chemicals to digest them, and then they used really pretty old school instruments to figure out which of these 4 bases were present and how much there was. And they found something weird. The bases A and T seem like they're always present in about the same amount, so those are the orange and red bars. And the bases G and C are always present in pretty similar amounts. What's going on with that? It seems like kind of a weird pattern. So that comes from the fact that those bases hydrogen bond with each other, and between 2 strands of DNA, there's always going to be these 2 bases hydrogen bonded with each other. So they're always going to look like this. If there's an A, it's hydrogen bonded to a T. If there's a G, it's hydrogen bonded to a C. So I probably blew past that a little too fast, but the 4 bases on the previous slide, they have these names: adenine, guanine, cytosine, and thymine, and people abbreviate those ATGC, AGCT, because those names are long. So we've got those names, ATGC, and that means on one strand of DNA, like this top line, we might have a certain sequence of those bases, A, G, C, T, etc. And on the bottom strand, it's going to have basically the opposite sequence. So T, C, G, A. And those guys are held together by these hydrogen bonds. So on a strand of DNA, we said we've got these individual nucleotides. A nucleotide up here, we've got P and S, and then C. So this would be one nucleotide with phosphate, sugar, and then this is the base cytosine. We've got another nucleotide, P, S, G, and on and on. So people call these pairs, like G and C, they call that one base pair. And we probably saw in the previous slide, A and T can form 2 hydrogen bonds together. G and C can form 3. So that's part of why A matches with T and G matches with C. They have these different structures, and these stick together really well. These stick together really well. Doesn't work so great if you try to match an A with a C, for example. So we've got on this picture that's actually duplicated, the A and the T have these 2 dotted lines for 2 hydrogen bonds between them. The C and the G have 3. And the DNA has this really characteristic shape that you've probably seen before, that people call a double helix. So we've got one strand of DNA and the other one is sort of matching it. It's kind of like a double spiral staircase. And people call these guys complementary strands because their sequences match up. So take a second and try to figure out which of these 4 possible sequences is complementary to this top sequence. And you can use the chapter 12 handout that's posted on Canvas to help you with this. So it has those pairing rules listed, but A always matches up with T and G always matches up with C, just like we saw here. Okay. Okay, so we've had a couple seconds to work on this one, and I'll reveal the answer. So we've got AGC, TAC, GATC. This is kind of like some of the problems from your homework. So if we match T with all the A's, we've got T, T, T. If we match A with all the T's, there's going to be an A and an A, etc. And we get that pattern, which looks like option D. So mark it. Yeah. Okay, very good. So that's pretty straightforward. That's not nearly as complicated as some of the stuff we've done in previous chapters. So that's nice. At this point, I'm going to switch over and look at basically what would have been the stuff I wrote on the whiteboard. So to give you guys sort of a picture of how this all works, this is sort of a summary. In biology, they call this the central dogma because it's like sort of the fundamental theory that they use to sort of think about what happens in a cell and how everything works. So let me kind of walk through this piece by piece since you're seeing it all at once instead of having it revealed one piece at a time. The idea is that DNA, deoxyribonucleic acid, is where all the information is stored. So that's like the code book for making all the stuff that makes up our bodies. And we said these vertical black lines are base pairs, and on one strand, every time we've got a base, a phosphate, and a sugar, that's one nucleotide. So if we've got 3 nucleotides, that's called a codon, and that's how your body is going to turn that into proteins. We'll look more at that next class. But DNA, before we can do anything with it, it's actually turned into RNA, which is really similar. It's ribonucleic acid, so the sugar is a little bit different in the backbone, and one of the bases is different. That process is called transcription. So that's when we take the DNA, we copy one strand of the DNA into RNA. RNA is always single-stranded like this, and RNA is a pretty fragile molecule. It falls apart pretty easily. So this is sort of like a temporary copy. The example I like to think of is that DNA is like a cookbook on a shelf. It's going to last a really long time, but whenever you want to go cook something, you can open it up and on a scratch piece of paper, write down the ingredients and the steps you're going to use. So the piece of scratch paper is like RNA. It's a temporary copy of the recipe you're going to work on. You can go into the kitchen, make that recipe, and then your little post-it note probably gets thrown away or something like that. Maybe it gets all stained and you don't care. So once you've got that temporary copy of the genetic code, the RNA, you can turn that into a protein. That process is called translation. So transcription basically means copying. If you are transcribing something, you're copying it. Translating is like you're turning it into a different language. The DNA and the RNA basically have the same language, but proteins are a whole different thing. So when we use the RNA sequence to make a protein, we're translating. We're turning it into a protein language. That's kind of a silly example, but we're making the protein that the DNA and the RNA both coded for. So one of these codons that was 3 bases in the DNA or the RNA creates one amino acid. So we've got sort of one codon worth of protein here. It's an amino acid residue. Once the protein is formed, it actually usually has a pretty complicated 3D shape, kind of like a ball. And if we zoom in on one little piece of the protein, this is kind of a funny cartoon, but you can imagine each little rectangle is an amino acid residue, and there's going to be thousands of these. A pretty small protein has a molecular mass of maybe 5,000, so there's still hundreds of amino acids in the tiniest proteins. They're pretty big. Awesome. So on that protein, we're going to have these R groups hanging off the side. That's going to be really important next class when we learn more about proteins. So I think I meant to show this picture earlier, but this is basically my version of the cartoon from your book that had the structure of DNA. And we've got the phosphate sugar backbone. I wrote little D's for deoxyribose. So phosphate deoxyribose is like the backbone of this protein. And then off the sides, there are these bases connected to all the sugars, and they're hydrogen bonded with nearby bases. If this base happens to be A, this one will be T. And if this were RNA, then instead of deoxyribose, this sugar would be ribose. And we already mentioned this a little bit, but we've got these attractions between 2 strands of DNA, right? Hydrogen bonds. So one strand of DNA is really long. It's a really big, long polymer. And this complementary strand is actually a separate molecule. They're held together by tons of hydrogen bonds, though, and so they basically act like one molecule. They're really stuck together because of having all these hydrogen bonds all the way along their length. So if we come back to our PowerPoint slide, we've got DNA and RNA. Now we kind of know how those work, right? DNA is our permanent copy of the information that the cell needs to survive. RNA is like the temporary scratch paper copy. And they're almost the same. They've got that one different sugar, deoxyribose versus ribose, in their backbone, which they're only different by one OH group. And then the bases, 3 out of the 4 are the same. So both of them contain adenine, guanine, and cytosine. But in DNA, there's this base thymine, which is replaced by uracil in RNA. So if thymine pairs with adenine, uracil can also pair with adenine. They're pretty replaceable. The only thing that's different between these two is this CH3 that points away from the other base. So these atoms are the ones involved in hydrogen bonding. So this CH3 doesn't really affect things. Okay. So this was This was a warm-up question from last year. It was in your learning journal, and I wanted to check out a couple of the questions that people asked related to this last year that I thought were good. One was, is it possible for a DNA chain to have a branch? Since a lot of the polymers we've learned about have branches. And my answer would be no, except for the fact that these all look sort of like branches whenever there's a base coming off to the side, right? It looks kind of like a branch. But other than that, nope, no branches. Do you guys have to remember what these bases look like? Definitely not. So all the structures that we've seen so far, like deoxyribose and those bases, those are going to be on your exam notes sheet, and they're in that handout that's posted on Canvas for this chapter. I'll basically give you all that information on your exam. No point memorizing that, so no way. Somebody asked, can we go more in depth into X-ray diffraction? So your book mentioned that's the way that they figured out the structure of DNA. So I'll go for 10 seconds into that. The way X-ray diffraction works is you start with some chemical and you want to know the 3D shape. You shoot X-rays at it. Some of those X-rays get bent, and if you have sort of like photographic film, you get a weird pattern like this one on the right. That doesn't really tell you anything yet. You have to do a bunch of math and computer stuff to figure out what that pattern means, but eventually you can figure out the 3D shape for a chemical. So this was sort of the first X-ray diffraction pattern that they used to figure out the actual 3D shape of DNA. All right, and the last thing somebody asked was, if nucleotides are the building blocks that make DNA, right? We said these are like the monomers in DNA. How do chromosomes relate to the polymer? Because you guys have probably heard that DNA in your cells is packed into these little pieces called chromosomes, right? So let's look at that on the next slide. That was a really good question. In your text, this blue stuff is just a quote from your text, basically saying one DNA piece is really long. It's maybe 6 feet long for one piece, for just the DNA in one of our cells. It's actually in a few pieces. That's like maybe 20, 25 molecules, 23 molecules. But the molecules are really, really long. So the question is, how do they fit into our cells? And what we're going to see on this page is that they need a lot of help to get shrunk down that far. This bottom text says, the DNA is compacted by wrapping around proteins. So this cartoon is sort of like just a strand of DNA. And there's some proteins in there, like this blue guy, that the DNA can wrap itself around to try to get a little more compact. If we zoom out, you've got DNA that's occasionally wrapped up, and then those wrapped up pieces the blue pieces can kind of stick to each other, so it gets even more compact. And if we zoom out, we've got this blue strand, which then gets sort of wound around this other protein that's shown in green. And then that gets wound back and forth along this other yellow protein. People call these scaffolding proteins. And eventually, we've got this pretty familiar shape of a chromosome. That's more what a chromosome looks like right when your cell is about to divide and turn into two cells. The left half is going to go to one cell, the right half is going to go to another cell. So normally chromosomes look a lot more like these vertical lines that have a little constriction in them. So that's one piece of DNA that's wrapped all around these proteins to make it much more compact. So each of these chromosomes that we see is really coiled up DNA. It's still this same molecule we've been talking about, but it's just all wrapped around these proteins to make it smaller. And they have these shapes kind of like this. All right, so this is called a karyotype. You don't need to remember that word, but that's sort of a picture like this of all the chromosomes inside a human cell that are organized. And it looks kind of like there's pairs, right? We've got a lot of them look like the ones with number 1 are both about the same size. The ones that are number 2 are about the same size. So we've all got 2 copies of each of these chromosomes, each of these 23 chromosomes. And so we've got 46 total. We've got 2 copies because you get one from each parent. And so when people are reproducing, the woman gives an egg, the man has sperm. Each of those has 23 chromosomes. And when those get together, that cell has 46, just like all the normal cells in a human body. So each of these, if we come back, each of these, where there's two that look the same, one's from your mother and one's from your father. And for the last pair of chromosomes, 23, you might have one that looks like this, where there's two that are identical, or you might have a big one and a little one. People call the bigger ones in this pair, in pair 23, X chromosomes, and they call the little one a Y chromosome, since a Y is kind of a little smaller than an X. It's made out of 3 lines instead of 4. It's sort of where people get that. Or because if you had these stuck together at the center to make that normal X shape of a chromosome, these guys would look like an X. This one would look a little more like a Y, since that top half is pretty small. All right, so those are called the sex chromosomes because they determine whether you're a female or a male. But things can go wrong sometimes in our cells, and we could end up with the wrong number of chromosomes. So this is the kind of thing that would only happen for a developing child, where inside the mother they might have some kind of a mistake where they get the wrong number of some chromosomes. If you had an extra copy of most of the chromosomes, if you had 3 copies of chromosome number 1 or most of these others, that person probably would not survive. But there's a few cases where they can. If you have 3 copies of chromosome 21, that results in Down syndrome, where the person survives, but they have some differences and seem physically different than most other people. So we already talked about this a little bit, but this is more info on the sex chromosome, this last one, number 23. And basically the default normal state for humans is being female, but if you've got a Y chromosome, there's some different stuff on there that makes you male, so it turns on maleness, if you want to call it that. All right, so when we've got DNA inside our cells, it looks kind of like this top part that we've been seeing. But when your cell is going to double, when it's going to reproduce and turn into a new copy of a cell, it has to get copied somehow. And the way that works is it's going to get unzipped. So it's going to get split in half into these 2 molecules. So all these hydrogen bonds are going to get opened. And a new piece of DNA is going to be synthesized or created inside the cell that matches each of those 2 strands. So you're going to start with one original strand, and the new ones are drawn in blue here. So you can copy that, and from each of those red original strands, people call those templates, you can make a new strand. So these 2 blue ones are new strands. And then we could do that again, and all the same information is contained within there. So there's special enzymes that unwind the DNA. They're shown right here. And they create a little open part of the DNA where it's not hydrogen bonded together. And then enzymes inside your body can move in these 2 opposite directions because they go from the 3' to 5' direction. So they can copy your DNA in opposite directions until they create 2 whole copies. So we've been talking about all this stuff related to DNA and what's its structure like. Now we kind of understand where the information in our cells is contained and stored, and we're going to use that to understand how gene modification works. But before that, I wanted you guys to see this other information. That's why I posted this story about breeding chickens. So before we had all the genetic technology that we have nowadays, we could use pretty old-fashioned methods to create animals and plants with the traits that we wanted. And I thought this story really illustrated that well. So in the last 75 years, they show the time it takes for a chicken to become mature and big enough to sell going way down from 110-ish days to less than half that long. So the chickens mature a lot faster. And it looks like here in the last maybe 40 years, they haven't been maturing much faster, but in that time, since 1980, they've been getting way bigger. So a chicken that's been alive for the same number of days has actually gotten almost 50% bigger, right? So nowadays, compared to in 1980, a chicken could grow the same amount of time, about 50 days, and be 6 pounds instead of 4 pounds. There's no fancy high-tech stuff going on here. They're just picking from the chickens the ones that seem the very biggest, that grow the fastest and make the most meat with the least amount of food, and they're breeding those with other chickens that have the same features. And so they get offspring that are like those and still have those traits. And so the chickens keep on basically getting better and better, the same way that dog breeds were made. So we saw before in chapter 11 that to make 1 kilogram of meat, chickens, compared to pork and beef, take way less food. So this is chickens, pork, and beef. And it takes way more food to grow 1 kilogram of pork or beef meat compared to chickens. Why are they so efficient? Well, a big reason is because they grow so quickly. So we don't have to keep feeding them every day for them to just sort of exist and live, right? I mean, think about how much food one of us eats in a day. Even if it's a lazy day and we're not doing very much, we eat kind of a lot of food. So the same thing is true for these animals. If you can get ones that grow really quickly, then there's less of that sort of food that just keeps them alive. And I just wanted to put the same picture back. It's basically the same information from chapter 11, but we saw that it takes a lot less food to raise those chickens. This is for broiler chickens, the same kind they were talking about in that article. If you haven't already read that, check it out. It's really interesting. And it's on the lecture 15 section on our page. So in nature, you guys have probably heard about this idea of natural selection, that idea that was first kind of put out there by Charles Darwin. So the idea is that in nature, processes can sort of select for certain features that help an organism survive. So maybe if there are 2 colors of beetles, orange and green, they would have equal chances of surviving. But if birds, like these guys, decide, ooh, we like the green beetles the best because they taste better, or they're easier for us to catch, or any reason, then that gives the orange beetles an advantage. And it might be a really small advantage, but over a long time, that advantage can kind of build up so that the orange beetles have a higher chance of reproducing and passing their genes, their DNA, onto their offspring. And eventually, it's almost all orange beetles because of that advantage, because the birds were much more likely to eat the green beetles. So this picture on the right is showing what's likely to happen in nature. A brown bug, this is kind of separate from the left picture, so one bug might not have any reason to pick bugs of a different color, light brown or dark brown or orange, but on a farm, like in the chicken example, we could say, let's put just dark brown bugs together so we have a really good chance of getting ones with the same features. Otherwise, the offspring bugs could have features from either parent. We might not know which color they're going to be. I already pretty much spilled the beans here. It's definitely option B. Definitely option B. So if evolution or adaptation is going on, it's not something that the organism has any choice in. It's just something that happens very slowly if some of them have an advantage. So speaking of breeding, this is another example kind of like the chickens. This wild mustard plant, if you found this and you were starving, you might not even realize you could eat it because there's so little edible material. But there's enough that a long time ago people figured out they could eat some parts of this, like the leaves, etc., and they started growing it. And some of them, maybe they noticed, ooh, this one has kind of an extra big flowering part. I'm going to breed it with this other plant like it that has an extra big flowering part. So they rubbed the flowers of one on the flowers of another to make offspring, and eventually they got broccoli. But actually, all of these plants that we eat, kale, broccoli, Brussels sprouts, cabbage, cauliflower, they all come from this wild mustard plant. So they're basically the same species, but different breeds, kind of like different breeds of dogs, like pit bulls and labs and collies. They're all the same species. We've just kind of selected for different traits. They're not different enough to be different species. If you guys haven't had kohlrabi, it looks kind of like an alien, but it is really good. It tastes kind of like the stem of broccoli after you peel it. Anyway, give it a shot. Okay, so this picture on the left, conventional breeding. That's sort of what we would do in the olden days, so in times before 50 years ago. We'd take one plant that we really liked, or animal, and we'd find some other version of it that had something we liked. So maybe it is resistant to a pest, or it survives better in cold weather. And that's because there's this piece of the DNA that makes a protein maybe that gives it an advantage. So we would mate those two together to get this offspring. They call it the F1 generation, and it would get one chromosome from each parent. Then it would have that gene, but it only would have a 50% chance of giving that gene to its offspring. So we would mate that again with our plant that we already maybe are growing on the farm. That's a really tried and true version. And we'd have some chance of getting that same gene still in there. And eventually, if we bred them enough and only selected for ones that we could tell had this feature we wanted, we could get one that basically had all the same genes that we wanted and also had that new gene that's colored in blue. What they're leaving out here is that 99% of the organisms that we produce this way wouldn't have the features we wanted. They might not have this gene. They might have other features, the red parts that are unwanted genes that maybe make if it's fruit, maybe make it have really small fruit or big seeds or a bad texture or a funny flavor. We're much more likely to get those because they make up a bigger fraction of the chromosome. So this is sort of the 1% that we have to filter out lots of garbage to get to. Lots of work shown here that's kind of not illustrated. So there's a couple other things that we could do in gene modification or genetic engineering. We could get the genes we want either from some other organism that's a totally different thing, or from basically the same species, but not the exact same version that we've got. So this right side is almost identical to the left side. We're saying, hey, we want that one gene. We'll copy that blue piece into this other little circular piece of DNA that we'll learn more about next week. We'll use that to put the piece we want directly into the new, or the kind of plant we already grow. So that's like super efficient. This middle version is really, really similar, but that gene we want doesn't necessarily have to come from the same species. So maybe there's some gene that makes the plant able to withstand cold temperatures, and it happens to come from a fish or something like that. It will work fine because the genetic code that we use or that plants use to make proteins, it's the same for all organisms. So you can swap stuff and it works perfectly fine. We're going to learn more about that in the next two classes after this one. So I want you guys to look, one sec while I look up the handout that I'm giving you and see if I can get you hard copies today. Okay, so there's a sheet on our lecture files page that sort of explains different ways that we can adjust the genetic material in an organism. I asked the administrator to print out some copies, so you guys, if you're in lecture right now in Olin 205, you should have some copies of that you can look at, but it's got a few different ways that we could change things. One is conventional breeding, so this thing we were talking about, just picking two examples that have traits we want and breeding those together until we get an organism just like we were hoping for. So the biggest dog or the smallest dog or the fluffiest dog or whatever. There's these other versions where we're using some kind of high-tech methods to just get one gene we want. And that's what people usually mean when they talk about a genetically modified organism, or a GMO. So it could be a plant, it could be an animal, could be bacteria, but it has altered DNA, and usually we've used some kind of modern genetic engineering where we just get one gene, one little piece of DNA to make it behave or look just like we like. So people sometimes talk about GM, or genetically modified material. So genetically modified organism. You could get more specific and say GM crops, GM fish, etc. So I've got a picture up here of these genetically modified fish, which are actually pretty cool. They glow different colors when UV light is shined on them. This is what the original fish looks like, and these are the GM versions of that fish. Check out that video that we posted online as part of these PowerPoint slides, but pretty cool. That's like a real product you could buy at a pet store. And this is saying a weed is any plant that we basically don't want. That sounds kind of random, but I've got an example coming up of some genetic modification. First, I want to go here and show you guys that file that hopefully you've got a copy of right in front of you right now. If not, you can download one. But this guy, please rotate. One sec, let me fix this. Okay, so this is that same sheet that you guys have. We could make plants or animals with the traits we want in a really slow process by breeding them with each other. So with chickens, we've made chickens that make more meat faster by just mating the chickens with the good traits together, and it's taken us, 75 or so years to get the chickens that make a lot of meat really fast. The other method that we saw on that PowerPoint slide a second ago is this one on the far right, transgenics. So we've got genes from somewhere else that we're putting into that plant or animal. RNA interference is another method. So this one in green. You guys are going to watch a movie before in a week, for our class in a week, at the beginning of week 10, about a new potato that uses RNA interference to change some of the way the DNA is expressed, the way the DNA gets transcribed and translated in the potato, but it doesn't actually change the DNA of the potato. It's pretty cool. And then mutagenesis, this is kind of a different thing. This doesn't count as a GMO. So the way this works is you take seeds and you shoot them with radiation to mutate them, to mess up the DNA. Or you soak them in toxic chemicals that also mess up the DNA. And random changes happen. So totally unpredictable, totally random. We don't even know afterward what has happened, really, a lot of times. But the resulting offspring that grow from those seeds are totally different. And that sounds crazy, right? That sounds like some dangerous new technology or something like that, but it's not. This is over 60 years old. It's where some of our favorite foods, like this seedless, red, really sweet grapefruit, the Rio Star, that's how it was made. Before that, grapefruit were not that sweet. They were not that beautiful red. They had more seeds. So this is weird because this sounds like a pretty Wild West, unsafe, perhaps, method where we don't even know what we're changing. These other methods, we really know what we're doing. So we're changing one or two genes or little pieces of DNA, little sequences that code for a protein, and we know exactly which ones they are. So it's kind of weird because this is a very controlled process, the two on the right, that are used to make GMOs. But people have a lot of fear and uncertainty about it, mostly because they don't really understand it. So let me pull our PowerPoint back up. Oh yeah. Sorry, one sec. Okay, so I had that thing on the last slide that said a weed is any plant that we don't really want, some unwanted plant. So we already saw that a GM crop has some kind of DNA that's been altered by genetic engineering. But before we can kind of think about a new version of corn, for example, we kind of need to know how it works normally. So normal corn, you take the seed, that's just like a kernel of corn that's matured, and you put it in the ground. You water it, you wait, and it grows into a corn plant. But there might also be some weeds, and those might use up some of the nutrients in the soil. They might mean that you end up getting less corn. So when you are ready to harvest the corn, you can harvest it, you can get some corn seeds off of there, replant them, and they'll grow into a new corn plant. This is all shown in black and white over here, but it's exactly the same as the left side. I just switched pens for some reason. So the new version, a few new versions of corn do almost the same thing. We're looking at sort of one brand name version, but there's GM corn that's made by Monsanto, and it's resistant to this pesticide, which has the trade name Roundup. It's called glyphosate or glufosinate or some name like that, and it kills lots and lots of plants. It's a weed killer. The way it works is actually, we understand it now probably. So this chemical inside plants, it stops this one enzyme that they use to make amino acids. So starting from the nitrogen in their fertilizer, so they take ammonia and they absorb it, maybe as nitrate or nitrite, but they absorb it and they turn that nitrogen and phosphorus that they also need in fertilizer, they turn those, I'm pointing at this chemical, but they turn the nitrogen into amino acids. And there's 3 of them that they really need. They can't get them by eating other organisms. They have to make the amino acids that they're going to use. This pesticide or herbicide, it keeps them from making those amino acids and they die. So this thing kills lots and lots of different plants. But there's new GM corn that is resistant to that herbicide. So this enzyme does not, or this chemical does not stop their enzyme from making amino acids. It has no effect on them. So if we use the GM corn, there's one other weird thing about it. That corn has, quote, Terminator technology seeds that only make one generation of plants. So we buy a seed for this GM corn, we plant it, we can spray this herbicide up here, this one in blue, all over the field, so no weeds grow. We've just got corn, no weeds. We go to harvest the corn, we get it, we plant the seed, nothing happens. The only way to grow a new generation of the corn is to buy a new seed from Monsanto. So this is something that a lot of people feel really strongly about in either direction. One reason people feel really negatively about it is they say that Monsanto is just being a bunch of greedy jerks. But you can kind of see why it makes sense maybe to have plants like this that can't reproduce, because if this corn plant reproduced and hybridized with some wild plant, then its genes could get into the wild plants and those would become immune to the herbicide also. So this sounds maybe kind of like a cool thing. One downside though that I've heard is you don't actually get more corn here than you would here up above with a regular corn plant. So this idea that the weeds are going to use up nutrients and take them away from the corn plant doesn't really seem to be super true or valid. So far, I don't see a lot of real benefits for society and the public in using this GM corn. It seems like mostly it was an idea, but it mostly so far seems that it just helps them sell more plants and more herbicides, since this one company sells both. We're going to actually learn, hopefully in a week, in lecture 17, about mosquitoes that they've engineered with this same basic idea. They can't reproduce, or they can't make viable offspring. So they're releasing these in places with Zika so that they will mate with females and then prevent viable offspring from happening, so that the mosquito population will go way down. I forgot to give you guys this question before, but option A I totally made up. It's really ridiculous. Option B isn't, well, I guess that's sort of true. That's what I was saying. There's pretty low chance of corn being able to breed with wild plants, but this really should make it impossible. C is true too, D is not true. So they have to put this in on their own. It's like one extra gene modification that they make when they're making the GM crop. Awesome. So now we really have a pretty good feeling for how DNA works. We've got a little bit of an idea for why we might make GMOs or how we might do it. So next class, we're going to look more at exactly how we turn DNA into proteins and how proteins work. And next week at the beginning of the week, we're going to see some pretty cool, interesting, specific examples of GMOs. This says there's a quiz, but that's a boo-boo. So we've got an extra class compared to last year coming up, lecture 18. So I should have just written lecture 18 next to that. I'll do it right here. Lecture 18. We're going to have a quiz, a review, and I'm making another exercise for you guys that I think will be fun. So I hope you guys have a lot of fun this week with your video sessions in lab, and hopefully I will see you soon.